Protein expression is a critical process that allows cells to create the proteins they need to function and thrive. Cells use the genetic information stored in DNA as a blueprint to manufacture specific proteins. These proteins then go on to perform a wide variety of important roles and tasks within the body. Understanding protein expression is key to understanding how living organisms work at a fundamental level. The image on this slide depicts an abstract representation of a DNA helix, highlighting the intricate structure and the flow of vibrant colors, representing the dynamic cellular environment where protein expression takes place. Protein expression is the process where genetic information in DNA is used to create a functional protein. This involves two key steps: transcription, where DNA is copied into messenger RNA, mRNA, and translation, where mRNA is decoded to synthesize a protein. Specialized cellular machinery like ribosomes and transfer RNA, tRNA, are essential for this protein synthesis process. Protein expression is fundamental for all living organisms, as proteins carry out a wide range of essential functions necessary for life. Proteins are the essential workhorses of cells, responsible for a wide range of critical functions. Understanding protein expression is crucial for elucidating these cellular processes and how they operate. A knowledge of protein expression is vital for developing new protein-based therapies to address various diseases and conditions. Protein expression is a cornerstone of bioengineering, enabling the production of novel proteins for applications like diagnostics and biomaterials. Exploring and harnessing the power of protein expression is key to unlocking new scientific and medical breakthroughs. Protein expression is a key step in many research and industrial applications. There are several different types of protein expression systems, each with their own advantages and disadvantages. Prokaryotic systems like E. coli are the most common, as they are easy to grow and can produce high yields of protein. Eukaryotic systems using yeast or mammalian cells can provide more accurate folding and modifications for complex proteins. Cell-free systems use cell lysates to synthesize proteins without living cells, allowing for rapid production and the ability to make toxic proteins. In vitro systems use purified components, offering flexibility and control, but can be more expensive and complex. Bacterial expression systems are a popular choice for producing proteins due to their simplicity, fast growth, and low cost. E. coli is the most commonly used bacteria for protein expression, but other bacteria like Bacillus and Pseudomonas can offer advantages for certain proteins. Bacterial systems are often used to produce large quantities of proteins for research, diagnostics, and industrial applications. The image on the right shows an E. coli cell, which is the most widely used bacterial expression system. Bacterial expression is a powerful and flexible tool for producing recombinant proteins at scale. Yeast expression systems are widely used for producing recombinant proteins. Yeast cells are eukaryotic, making them suitable for expressing complex proteins with proper folding and modifications. Yeast are relatively easy to grow and genetically engineer to express high levels of target proteins. Common yeast species used include Saccharomyces cerevisiae, baker's yeast, and Pichia pastoris, methanol yeast. Yeast systems offer advantages like efficient protein production, cost-effectiveness, and scalability. These features make yeast a popular choice for research and industrial applications. Mammalian expression systems use mammalian cells to produce proteins. These systems are valuable for making complex proteins that need post-translational modifications like glycosylation to function properly. Mammalian cells provide a natural environment for protein folding and modification, leading to greater stability and activity compared to other systems. The image shows a mammalian cell culture glowing green under a microscope, with a complex network of cells in a petri dish. The green glow and soft lighting create an appealing visual to illustrate the mammalian cell culture. Insect cells are a widely used system for producing recombinant proteins due to their ability to efficiently express complex proteins with proper post-translational modifications. The baculovirus expression system is a common technique used to leverage the capabilities of insect cells for protein production. Insect cell expression offers several key advantages, including high yields, cost-effectiveness, and the ability to produce proteins with the necessary modifications for their intended applications. These systems are highly scalable, allowing for large-scale production of recombinant proteins for use in therapeutics, diagnostics, and research. Cell-free protein expression is a powerful technique that allows us to produce proteins outside of living cells. This approach eliminates the complexities of cell growth and protein folding that can be challenging when working with living systems. Cell-free systems offer significant advantages in terms of speed, flexibility, and the ability to synthesize proteins that may be toxic or difficult to produce in living cells. The image on the slide shows a close-up view of a petri dish with a blue liquid, suggesting the cell-free protein expression is taking place in a controlled, laboratory environment. I'll highlight how this technology is enabling new breakthroughs and opening up exciting possibilities for protein engineering and production. The gene sequence, including the specific codons used, can affect the efficiency of translation and the stability of the final protein product. A protein structure, including how it folds and any post-translational modifications it undergoes, plays a key role in determining its expression levels. The cellular environment, such as temperature, pH, and nutrient availability, can significantly influence the rate of protein synthesis. The choice of host cell, whether it's a bacterial, yeast, or mammalian system, can have a major impact on the overall level of protein production. Codon optimization is a powerful technique to enhance protein expression by using codons preferred by the host organism. By adapting the gene sequence to match the host's translational machinery, we can significantly boost protein synthesis efficiency. The process involves substituting codons while maintaining the amino acid sequence, resulting in more efficient translation. This allows us to fine-tune the gene for optimal expression in the target host. The goal is to maximize the expression of the desired protein by leveraging the host's natural codon biases. Promoter selection is a crucial step in optimizing protein production. We want to choose a promoter that has the right balance of strength, specificity, inducibility, and compatibility with the host cell. Strong promoters drive high levels of gene expression, which is important for producing sufficient amounts of the target protein. The promoter should be specific to the host organism and the desired expression level, without activating unwanted genes or interfering with cellular processes. Inducible promoters allow us to control expression of the target gene, enabling optimization of conditions and preventing toxicity from overexpression. Finally, the chosen promoter must be compatible with the host cell machinery, including transcription factors, translation factors, and ribosome binding sites. The selection of the right host cell is critical for achieving optimal protein expression. Key factors to consider include the size, structure, and post-translational modifications required for the target protein. The host cell must be compatible with the expression system used and have a high capacity for protein production. Other important considerations are the host cell's genetic background, growth conditions, potential for protein modifications, as well as cost, safety, and regulatory compliance. Carefully evaluating these factors will help ensure successful protein expression. Chemical inducers like IPDG are powerful tools to trigger the expression of target proteins in our system. Temperature shifts, such as a heat shock, can also be used to induce protein expression. This can be a useful alternative approach. Carefully controlling the timing of induction is crucial for optimizing the yield of the target protein. We need to find the sweet spot. Combining different induction methods, like using both chemical and temperature triggers, can be an effective way to enhance overall protein expression and production. Protein folding is the process by which a protein structure assumes its functional 3D shape from a linear chain of amino acids. Post-translational modifications are chemical changes made to proteins after they are synthesized, which can alter the protein's function, stability, location, and interactions. Key post-translational modifications include glycosylation, adding sugars, phosphorylation, adding phosphate groups, and acetylation, adding acetyl groups. These modifications are essential for proper protein function and are often highly regulated, playing important roles in cellular processes like signaling, regulation, and trafficking. The slide provides an overview of protein folding and some common post-translational modifications. We'll dive deeper into each of these topics in the following slides. Protein purification is a crucial step in many research and industrial processes. There are several key techniques used to purify proteins, each with their own advantages. Chromatography separates proteins based on their physical and chemical properties. Ultrafiltration uses a membrane to separate proteins based on their size. Centrifugation separates proteins based on their density using centrifugal force. Electrophoresis separates proteins based on their charge and size using an electric field. Each of these techniques has specific applications and can be used alone or in combination. The choice of technique depends on the properties of the target protein and the desired level of purity. Affinity chromatography is a powerful technique that exploits the high affinity between a target protein and a specific ligand attached to a stationary phase. The target protein binds to this ligand, while other proteins pass through the column. This allows for highly selective purification of the target protein. To elute the bound protein, we use a solution that competes with the ligand or alters the binding conditions, causing the target protein to be released from a column. This technique offers excellent specificity and is a crucial tool for purifying and isolating proteins of interest. Ion exchange chromatography is a powerful technique for separating proteins based on their net charge. The stationary phase contains charged groups that interact with oppositely charged proteins, allowing separation. There are two main types: cation exchange, which binds positively charged proteins, and anion exchange, which binds negatively charged proteins. Bound proteins can be eluted by increasing the salt concentration or changing the pH, which alters the protein's charge and reduces its affinity for the stationary phase. This technique has many important applications, including purifying proteins, antibodies, enzymes, and other biomolecules, as well as desalting and buffer exchange. Size exclusion chromatography is a technique that separates proteins based on their size. Smaller molecules can pass through the porous beads of the stationary phase, while larger molecules cannot enter the pores and elute first. The elution profile shows the size distribution of proteins in the sample. Different sized proteins will elute at different volumes. This technique provides information about the molecular weights and size range of the proteins present. Western blotting is a powerful analytical technique used to detect and quantify specific proteins in a sample. It involves separating proteins by size using SDS page, transferring them to a membrane, and then using antibodies to identify the target protein. This technique is widely used for a variety of applications, including studying protein expression, protein-protein interactions, and detecting biomarkers in clinical samples. The process is composed of several key steps: protein separation, transfer to a membrane, blocking, antibody incubation, and signal detection. Mass spectrometry is a highly sensitive analytical technique that allows us to identify and quantify molecules based on their mass-to-charge ratio. It works by first ionizing the molecules, then separating them based on their mass-to-charge ratio, and finally, detecting their abundance. This technique is widely used in proteomics, the study of proteins, to identify and quantify the proteins present in a sample. Mass spectrometry provides very detailed information about the molecular composition of a sample, making it an invaluable tool for researchers and analysts. Our recombinant proteins are a crucial part of modern medicine, allowing us to treat a wide range of diseases like cancer, diabetes, and autoimmune disorders. These therapeutic proteins act as drugs, replacing or enhancing specific functions in the body that are not working properly. A recombinant protein technology has also revolutionized the production of enzymes used in many industrial processes like food processing, biofuel production, and detergent manufacturing. A recombinant enzymes offer advantages over traditional methods, such as higher yields and improved stability. The ability to produce these important proteins and enzymes through recombinant technology has been a major advancement in both medicine and industry. Therapeutic proteins have revolutionized disease treatment, offering targeted and specific therapies. Recombinant DNA technology is a key enabler, allowing expression of desired genes in suitable hosts. Therapeutic proteins encompass a wide range of molecules like antibodies, enzymes, hormones, and growth factors. These proteins are used to treat various conditions such as cancer, autoimmune diseases, infections, and genetic disorders. Biotechnological advancements have been crucial in enabling the production and therapeutic use of these proteins. Enzymes play a vital role in various industrial processes, catalyzing specific reactions and improving efficiency. Examples include proteases in detergents for stain removal, lipases in biodiesel production, and amylases in brewing. Recombinant protein expression has revolutionized enzyme production by introducing the desired enzyme gene into a host organism. This approach enables the production of enzymes with improved properties and higher yields compared to traditional methods. Recombinant expression offers significant advantages over conventional enzyme production techniques. Structural biology is the study of the 3D shapes of proteins, which is crucial for understanding how they function. Knowing the structure of a protein can help us determine how it interacts with other molecules, which is vital for drug discovery. Structural biology also plays a key role in biotechnology, as it allows us to design new enzymes and other proteins with specific functions. Understanding protein structure is essential for advancing our knowledge in fields like medicine and biochemistry. Protein engineering is a powerful field that allows us to create new proteins or modify existing ones to have desired properties and functions. There are three main approaches to protein engineering: rational design, directed evolution, and de novo design. Rational design involves making targeted changes to protein sequences based on our understanding of protein structure and function. This allows us to engineer proteins with specific, desired traits. Directed evolution uses random mutagenesis and selection to create libraries of protein variants from which the best performing proteins can be selected. This mimics the natural process of evolution. De novo design is the most ambitious approach, where we use computational methods to design completely new proteins from scratch, predicting their structure and function. Each of these methods has its own strengths and can be applied depending on the specific goals and challenges of the protein engineering project. Protein aggregation is a common challenge in protein expression, leading to reduced yields and difficulties in purification. This can be addressed through optimization of expression conditions and the use of solubility-enhancing tags. Expressed proteins can sometimes be toxic to the host cells, hindering their growth and overall production. Strategies like using inducible promoters or exploring alternative host systems may help mitigate this issue. Scaling up protein expression processes can be challenging, requiring careful optimization of parameters like media composition, bioreactor design, and process controls. Thorough process development is crucial for successful scale-up. Regulatory considerations are particularly important for proteins intended for therapeutic applications. Strict guidelines must be followed regarding safety, quality, and manufacturing practices. Close collaboration with regulatory authorities is essential. Protein aggregation is a major challenge in protein expression, leading to decreased protein yield and functionality. Misfolded proteins can aggregate, forming clumps that can interfere with cellular processes and product stability. Strategies to mitigate protein aggregation include optimizing expression conditions, using chaperone proteins, and modifying protein sequences. These strategies can help improve protein solubility and stability, leading to higher yields and better product quality. Recombinant protein expression can be a burden on the host cell, leading to cellular stress and toxicity. Overproduction of a foreign protein can disrupt the cell's normal metabolic processes, resulting in toxicity. Misfolded or aggregated proteins can induce toxicity and interfere with the cell's normal functions. It's important to carefully manage protein expression levels and folding to minimize the risk of cellular toxicity. Strategies like optimizing expression conditions, improving protein folding, and reducing metabolic burden can help mitigate toxicity issues. Scaling up protein expression is crucial to meet high demand for therapeutic proteins, enzymes, and other recombinant products. Large-scale production must be efficient and cost-effective to make recombinant protein technologies commercially viable. Optimization of fermentation, cell culture, and purification processes is essential for scalable and robust production. Scaling up requires larger bioreactors, purification systems, and specialized facilities to handle large volumes of material. The FDA plays a critical role in regulating the manufacturing and distribution of therapeutic proteins in the U.S. They set strict standards for purity, potency, and safety that must be met before a protein can be brought to market. Good manufacturing practices, or GMPs, are the guidelines that ensure the quality and consistency of manufactured protein products. GMPs cover every aspect of production, from the raw materials all the way through to the final packaged product. Adhering to GMPs is essential for meeting the FDA's regulatory requirements and getting approval to sell therapeutic proteins. Automation and high-throughput screening will streamline protein expression workflows, enabling rapid testing of different expression conditions. Advanced computational tools will play a crucial role in designing novel proteins with enhanced stability, function, and expression. Synthetic biology will enable the creation of innovative protein expression systems with tailored properties to meet specific needs. Precision protein engineering will allow for the design and production of proteins with targeted functions and characteristics.